Everybody here? Anyone not here? Okay, I'd like to welcome everyone to the first full day, second session. I'm Shelley Berger, welcoming you with Bob Kingston and Jörg Mueller. Jörg? Okay. And it's my great pleasure to introduce to you Fred Winston, who's our second keynote this morning. He needs no introduction to maybe the first few rows, I guess the old farts, as Bob referred to us. Those of us that, well, maybe half of the auditorium. Fred needs no introduction. However, because he's a luminary in the field. To the grad students and postdocs here to learn, I say listen up. Fred's truly had a career trajectory that is worthy of the career planning session that's coming up in a couple of days. So that's fantastic. Now, let me tell you about his career. It certainly helps to start out as an undergrad at University of Chicago, do your PhD at MIT, and then postdoc at MIT and Cornell. Fred then, during that time, made amazing choices. He worked with three of the great geneticists, yeast geneticists, Mark Rose, David Botstein, and Jerry Fink, great yeast geneticists. He then, in 1983, became an assistant professor at Harvard. Now, Fred, I think, is an exemplar of Louis Pasteur's famous comment, where observation is concerned, chance favors only the prepared mind. I think Fred noticed while he was working with Mark seemingly a simple, trivial observation of the time that an insertion of a transposable element into a selectable gene or a nutritionally required gene in yeast would cause the yeast not to grow. And he reasoned that looking for suppressors of this transposable element insertion would yield interesting suppressors. And indeed, he found many interesting, really wonderful transacting regulators that really span the gamut of transcription and chromatin. So, for example, he found one of his first papers was on SPT3, which, along with I didn't say, so the name of the suppressors are SPT, suppressor of transposable element, and he found that SPT3 and later SPT7, SPT8, and SPT20 are all components of SAGA, near and dear to my heart. It has the GCN5 coactivator and acetyltransferase. He found SPT15, TBP, Tata binding protein, for those of you who love it. For those of you that love elongation, SPT4, 5, and 6, elongation factors, and later found that some of these factors have a role in suppressing cryptic transcription. He found that there are intragenic transcripts, one of the first to find regulatory roles of intragenic transcription, all using these sorts of approaches. I'd say for this field, my favorite observation from Fred's is a 1988 paper that he published soon after he started his own career, and a beautiful study where he found that that mutations in histone genes act as SPT suppressors of this transposable element insertion. He showed that changes in histone dosage can suppress this insertion, and thus found really the first real true observation that chromatin regulation is involved in transcriptional regulation. All right, so I want to end with a remark that in a time of really challenging funding, I think Fred's work exemplifies the enormous importance of model systems, working in yeast, Saccharomyces cerevisiae and then Schizomyces pombe, he's really shown, really through all of his findings, conservation of these pathways in his own immortal words in larger eukaryotes. So, Fred?
Thank you, Shelley. I want to thank Shelley and Bob and Jörg. Of course, it's a huge honor to be invited to give this talk, and I really mean that. As Shelley said, I'm going to talk about our studies in transcription in yeast, and that's yeasts are shown here. We study Cerevisiae and Pombe, and today I'm going to talk mainly about studies in Cerevisiae with a tiny bit of Pombe right at the end, and I'm going to talk mainly about one of the SPT proteins that Shelley mentioned, SPT6, but then a little bit about one of the others, again, right at the end of the talk. So let me start by introducing you or reviewing for most of you some aspects of transcriptional control shown on the next slide. So as everybody knows, there are a huge number of factors involved in transcription initiation. This is a typical yeast gene, except that I'm showing a TATA, which is not at most genes. But there are a large number of factors, site-specific DNA binding activators, coactivators, general factors, TBP, RNA polymerase II. Upwards of 50 proteins are required for specific and regulated transcription initiation. And of course, there's an equally large number of factors required for transcription elongation once polymerase moves down the template. And of course, as we all know, this occurs on a chromatin template. Here's a high-resolution view of nucleosomes that lie across coding regions and regulatory regions, and these are barriers that have to be dealt with by the transcriptional machinery for normal gene expression. Now, something that I found quite surprising, I think the field found surprising, starting several years ago, is that it's actually a number of elongation factors that are required for the integrity and accuracy of transcription and transcription initiation as well. So I want to give a little bit more information about elongation factors, and that's shown on the next slide. All right, so now polymerase has moved down. This shows some, but certainly not all, of the factors that move along with polymerase. These are all conserved, as shown down here. Shelley mentioned hard times for funding. If you would like your picture up here instead of Darwin's, I will do it for a small contribution to my lab. Okay, so templates in general, histones towards the 5' end of the gene are methylated on histone H3 lysine 4 across the rest of the body of the gene on lysine 36. As I said, there are a large number of factors, and I think it's quite interesting because many of these are essential for viability, meaning that it's not a lot of proteins contributing to one function. Each one seems to carry out a vital function. Every one that has an SPT name is essential for viability, as well as this one, IWS1. Now, we've been studying a lot SPT6 over the last several years, and as I said, that's what I'm going to talk about today. But we're interested in all the ones that are in yellow, and we're studying those in the lab, how they relate to SPT6 and elongation. I'll say a word about SPT5, tiny bit about SET2. I won't talk about IWS1 today, also known as SPIN1, but we're studying that one as well. So here's an overview of what I'm going to talk about today. I was asked by Bob and Shelley to give my own brief history of SPT mutants, so I'll tell you a little bit of what you already heard from Shelley. Then I'll give you some more introduction to SPT6. I'll spend the bulk of the talk focusing on SPT6 control of transcription, meaning our analysis of what happens to transcription in SPT6 mutants. And I'm going to start with some very old results, almost 10 years old, and I'm going to go up to some very new results where we haven't really had time to analyze all the data. In a lot of cases, including some of the old results, we don't understand what's going on. So I'll welcome any input from anybody. And then at the very end, as I said, just a brief view of our initial analysis of SPT5. So, brief history of SPT mutants. It actually started here at Cold Spring Harbor. I had been a postdoc in Jerry Fink's lab for a year at Cornell, and it started on two exceedingly high-risk, long-shot projects, and not unexpectedly, neither of them worked. So after a year, I said to Jerry, someday I'd like to get a job other than being a postdoc. Maybe I should switch projects. And he suggested, since we were about to come to Cold Spring Harbor for the Yeast meeting, that we wait till we're there and then find some time to sit down and talk about possible projects. So we found time when we actually met in this building. For those of you not familiar with this old use of the word library, it's a large building that contains books and journals. And so we met inside this building and had a discussion about what to do as the next project. And so we decided to focus on yeast transposable elements, and the ones of interest are shown on the next slide. So in the Fink lab, before I got there, they had isolated a spontaneous insertion mutation caused by a yeast TY1 element. Studies of eukaryotic transposons was very new at the time, that had integrated into the 5' non-coding region of His4. And then they had isolated a derivative of it that had undergone recombination between the long terminal repeats, so it had just this solo LTR or delta sequence upstream of His4. And what was known was that these insertions altered normal expression of the His4 gene at the transcriptional level. So we decided to do a mutant hunt, as Shelley said, and we started with this insertion mutation of just the delta, His4 912 delta, and that's shown in a little bit more detail on the next slide. All right, so here's the wild-type His4 gene as a reference. Here's the insertion. This is not to scale. It's actually 334 base pairs long and has its own promoter. In this mutant, transcription initiates rather than at the His4 start site in the delta or LTR start site. It makes a longer than normal messenger RNA that's non-functional for translation of His4 due to upstream translation, initiation, and termination codons in this region. So we decided to select for His plus suppressors, and I think Shelley cast too much rational thought on the process because we didn't have the faintest idea what we were going to get. Really, the driving hypothesis of the work was maybe we would find some interesting mutants. We didn't know if we were going to get excision of the LTR, if we were going to get transposon-mediated rearrangements, or something else. And it was only in hindsight that we really understood we were studying transcription, and that came from looking at all the suppressors that we got and showing that they all shifted transcription at some level back to the normal start site, at least as judged by northern analysis. And so this identified about 20 SPT genes, and we've been studying them over many years now. And I should say this was a very, very rich time for yeast genetics. There were many interesting mutant hunts for transcription mutants at the time, SRB mutants in Rick's lab, ADA mutants work done by Shelley when she was in the Guarenti lab, switch SYN, SNF mutants in the Carlson and Herskowitz and Naismith labs. Many interesting transcription mutants were coming out at the time. So as we learned more and more about SPT genes and the proteins that they encoded, we learned that they affect many different aspects of transcription, and that's shown here. Everywhere you see an SPT, there's a protein in that complex or type of factor that was affected by mutations. The only case that didn't come out of the kind of mutant hunt I told you were the ones in RNA polymerase. Those were done by site-specific mutagenesis of RPB1 and RPB2 by a grad student in Rick Young's lab many years ago. So there were many classes of SPTs, but many of them fell into two classes, and Shelley already told you about some of these, but I just want to say another word about them, and they're listed on the next slide. So even though they were selected as suppressors of insertion mutations, of course we could test them for many other phenotypes and put them into classes based on the mutant phenotypes. So we had class 1 here, and as Shelley mentioned, SPT15 we found encodes TBP back at a time when nobody had cloned the gene yet. We learned this from a phone conversation I had with Steve Hahn long time ago. Back then, it wasn't that we just emailed DNA sequence. Just actually, over the phone, I read to him a restriction map, and we confirmed that we had the same gene. He had cloned his starting with the protein. We had cloned ours starting with a mutant. All right? And 4 of the other SPT genes turned out to encode proteins that are part of SAGA. This is a fabulous collaboration with Jerry Workman, Shelley, and Dave Alice labs. And so based on this knowledge that had SPT, ADA, and GCN5 proteins, in a phone conversation with Jerry Workman, he said, Fred, we need to name the complex so people remember it. So we thought about this, and creative souls that we are, the best we could come up with was SAG or gas. So phone conversation ended. I go to lab meeting with Brad Cairns, who was a postdoc in the lab at the time. I brought this problem to lab meeting. Brad Cairns, who was sitting there, immediately said, why don't you add an A for acetyltransferase? So creative with acronyms, as he is in science, the name SAGA was born. And so you can thank Brad for me not talking about the SAG complex. All right, class 2, again, many of the proteins were identified in other ways. Shelley mentioned in collaboration with Marianne Osley's lab, We showed that these two encode histones. This is some of the early evidence that chromatin structure actually controls transcription. Hiroshi Honda's lab purified the mammalian equivalents of SPT4 and 5 in a factor called DSIF. Danny Reinberg's lab purified FACT, identified and purified FACT, and showed that one of the protein components is SPT16. Another very interesting aspect of all of this is that mutations in all of these genes we found a long time ago suppress switch-SNF mutations, and that was one of the early genetic connections between switch-SNF and chromatin remodeling complexes, something that was confirmed biochemically by Kingston, Green, and Crabtree labs studying mammalian switch-SNF, and Peterson and Workman labs studying yeast switch-SNF. Okay, so that's my overview of the SPT mutant hunt. So now let me get back to SPT6, the main topic for today, and introduce you more to SPT6. So it's a very large protein, has many domains. The two that we're most interested in, or we know the most about, is at the amino terminal end, there's an acidic region that binds to nucleosomes and histones. This is work from our lab, Chris Hill and Tim Formosa. And several labs showed that the two SH2 domains at the C-terminal end are required for SPT6 to interact directly with the CTD of RNA polymerase II largest subunit. And this has been shown by many labs. I should point out that these are the only two SH2 domains in the entire yeast proteome for either Cerevisiae or Pombe. So we know that SPT6 directly interacts with histones in Pol2. It co-localizes with RNA polymerase II across transcribed regions, mainly transcribed regions and not regulatory regions. And this helped give rise to the view that SPT6 is involved in transcription elongation. And finally, it controls transcription, which I'm going to talk about today, chromatin structure and histone modifications. Depending which yeast we're talking about, it can be required for H3K4 methylation, H3K36 methylation. There's some evidence in mammalian cells that it's required for normal levels of H3K27 methylation. So just because I'm not going to talk about chromatin structure or histone modifications, I'll just show you one example of an experiment shown over here. This is an NET-seq experiment we did in Pombe, comparing a wild-type strain to an SPT6 mutant. This is a metagene analysis for wild-type in black. You can see the typical nucleosome depleted region, and then peaks for the +1, +2, +3, et cetera, nucleosomes. In the SPT6 mutant, we see that this is all blurred out, that we see weaker peaks and they're slightly further apart, and we conclude that chromatin structure, this and other evidence, lets us conclude that chromatin structure is largely in disarray in SPT6 mutants. So because of these interactions and because of the phenotypes we see, we think SPT6 has multiple direct effects in vivo. So what I want to talk about today is what classes of transcripts are regulated by SPT6. And we're interested in this because we think it informs us what SPT6 does, but it also informs us about possible transcripts that are made and their possible roles in vivo. So in other words, what's the potential for transcription across the genome? So I want to start with intragenic transcription, which Shelley mentioned in the introduction, and that's an example of that shown on the next slide. So here's an old Northern analysis looking at two genes, FLO8 and sterol11. And I want to point out before we go there is that almost all the experiments I'm going to show you were done in this mutant allele, SPT61004. It's a deletion of this helix hairpin helix motif. It's temperature sensitive. So we look at strains after a shift to non-permissive temperature for 85 minutes. Protein levels drop to about 20%. The cells are still alive. So we view it as a depletion of SPT6. So that means a wild-type strain or a mutant strain, both grown under the conditions I just described. In wild-type, we see a full-length RNA. In the mutant, a shorter RNA. And so from this type of analysis and from other analyses, we conclude that there's transcription beginning intragenically across the 3' portion of the gene, missing the 5' end. Now, if you remember back a few slides ago, this is really similar to what we saw in the SPT mutant hunt. In both cases, we have an upstream promoter that's functional in the wild-type situation and a downstream, normally silent promoter that now gets activated in the SPT6 mutant. So we can begin to understand why we got these mutations, although we don't really understand the mechanism yet of why they happen. So some more information about intragenic transcription. It's widespread in SPT6 mutants. We knew from microarrays done in collaboration with Tim Hughes' lab that it occurred at least close to 1,000 genes. Now we think it's even wider spread. I'll talk about that in a minute. We think it can be enabled by many types of changes in chromatin structure, nucleosome loss, or changes in histone modifications. So we were interested in understanding intragenic transcription better. All of the analyses we did merely identified transcripts across the 3' portions of genes, and in only 1 or 2 cases had we actually shown that they were due to new initiation events. So we wanted to look at this more carefully and at higher resolution, so we did it using a method called TL-seq, and that's introduced here. I'm not going to describe the method. It's a method developed by Wendy Gilbert's lab at MIT. It's similar to a method called CAP-seq, published by Craig Mello's lab. And in this method, one builds a deep sequencing library of fragments purified based on their having a cap on the messenger RNA and sequencing across the 5' end of the RNA. So we did that for comparing wild type and SPT6 mutants, and a summary of the results is shown here. So we found a very large number of changes in an SPT6 mutant. We found about 7,000 new initiation peaks on the sense strand and about 2,000 on the antisense strand. And down here shows some more of the data. You can see if we compare on the right a wild-type strain to an SPT6 mutant, there's a very poor correlation for transcription start sites. It's very different across the genome. But just to show that the method is reproducible, if we compare 2 separate biological experiments looking at an SPT6 mutant, we see that the data are highly reproducible. So we think that there are vast changes across the genome in transcription initiation in an SPT6 mutant, many of them beginning intragenically, normally silent, cryptic, but then in an SPT6 mutant they become activated. So this gives rise to the question, I want to show you a couple of specific examples. These are the 2 genes where I showed you northerns before, FLO8 showing being transcribed right to left. Here's the normal start site, wild type's on the top, SPT6 is on the bottom. And you can see in the SPT6 mutant, here's the intragenic start. There may be a couple other weak ones. Now sterile 11 shown left to right. Here's the wild type start, and here are the intragenic starts that we see. We take some hope from this little peak here that maybe this is also made at a low level in wild type strains. So why are there intragenic transcripts? All right, a couple possible models, not mutually exclusive. Intragenic promoters must be repressed to prevent aberrant transcription. Normally, the cell wants to keep them off at all costs because otherwise it will mess up the normal gene's transcription. Or second possibility is that intragenic promoters actually have a biological role and are activated under some conditions. And I think there's evidence for both of these that I don't have time to describe today. But if the second one is true, then we expect that some of these RNAs would be translated into proteins, and that turns out to be true, and some examples of that are shown on the next slide. All right, so we actually screened by brute force 146 possible cases where proteins might be made and found detectable proteins made in 20 of those 146 cases. This is Western analysis comparing wild type to SPT6 mutants. We see proteins made corresponding to the ATG on the shorter RNA. And we did some controls to show that these aren't merely protein breakdown products. So some RNAs are translated, and we've shown recently that some are translated even in alternative reading frames to the full-length transcript. So just to summarize about intragenic transcription, these promoters exist throughout eukaryotic genomes. There was a recent paper from the D.L. Meda lab in Lisbon that intragenic transcription is widespread in mammalian cells as well. In SPT6 and other mutants, this allows extensive expression of novel genetic information, both at the level of RNA and of protein. So some non-coding probably, and some coding. Wild-type strains also express some of this information under certain growth conditions. We showed a few years ago that under nutritional stress, some intragenic promoters are activated. A paper from Gloria Brar and Jonathan Weissman showed that this happens during meiosis in yeast. A recent paper from the Wang lab at Florida State shows that it also happens under DNA replication stress. And there are multiple cases in mammalian cells where intragenic starts encode functionally distinct proteins. So we think that this is an emerging area, and we're very interested, and we are pursuing it further. Okay, now the TL-seq experiment that measured start sites also told us something about transcription initiation at normal start sites that was somewhat surprising. We knew that there was some effect of SPT6 on normal initiation from work from Jessica Tyler's lab looking at FO5 and from our lab looking at CHA1, but we had never looked at this in a systematic way, partly because the localization across coding regions and lack of localization in regulatory regions gave us the bias that it probably wasn't affected widespread. Turns out we were wrong, and that's illustrated here. Here's an example of 2 genes from our TL-seq analysis, CHA1. You can see in the SPT6 mutant, there's a higher level of initiation than in wild type. Here's a second case, TKL2. This gene is basically silent in a wild type strain. Now, high level of initiation in an SPT6 mutant. And of all the genes we were able to assay from our TL-seq experiment, about 1/4 of them were increased in level 2-fold or more in an SPT6 mutant. So we find this very interesting, still a little mystified about how it works, but it's an area that we will be investigating further. Okay, now finally, we were interested in asking, what's the level of synthesis of all of these RNAs? Intragenic, SPT6 affects antisense transcription, which I'll be talking about, and other RNAs, because it had been shown in many organisms, in Pombi and Cerevisiae, and mammalian cells, that some of these RNAs are present, are normally unstable, and are only seen when they are stabilized in exosome mutants or other kinds of mutants. So we wanted to look at transcription where degradation and stability was taken out of the picture. So for that, we turned to another method called NET-seq, developed by Sterling Churchman and Jonathan Weissman. I'm not going to go into detail, but basically in this method, for yeast, one immunoprecipitates RNA polymerase II. It will co-purify with nascent RNAs. Then we build a sequencing library to sequence in from the 3' ends of the RNAs. And so the first base tells us the active site of RNA polymerase II on either the sense or the antisense strand. And an example of that is shown here. Here's the TKL2 gene. I just showed you it's expressed in SPT6 mutants, and it's transcribed from right to left here. You can see in wild type a very low level of Pol2 across the gene, as opposed to an SPT6 mutant, a much higher level. Now let me show you another gene on the next slide, DDC2. This is a gene where we had shown there's intragenic transcription a long time ago by northern analysis. And now if we look by NET-seq on the sense strand, going from left to right, wild types on the top, SPT6 mutants on the bottom, we see a higher concentration of active Pol2 across the 3' end, consistent with these transcripts that we see. Now, in addition, I said we knew that there was a lot of antisense transcription going on in SPT6 mutants. We wanted to know if that was due to increased synthesis, so we can look at the antisense strand as well, and that's shown here. So now antisense is being made from right to left. We can see in the wild type there's a very low level of polymerases, but in the SPT6 mutant, it's greatly increased. So of course we wanted to know how widespread is antisense transcription in SPT6 mutants. We knew from Pombi it was pretty widespread, and we also knew that it was fairly widespread in other mutants as well. So we looked genome-wide, and then the first thing I'm going to show you is a comparison to a previously well-characterized mutant, CET2, well-characterized for antisense transcription. All right, CET2 encodes the H3K36 methyltransferase in yeast, initially identified and purified by Brian Strahl. This lab still works on it. CET2 deletion mutants have extensive antisense transcription. This has been characterized in Jerry Workman's lab. It was known that SPT6 mutants have greatly reduced levels of H3K36 trimethylation, initially shown by Jane Meller's lab. Also the Workman, Strahl, Pralick, and our labs have also studied this. So we compared CET2 and SPT6 and looked at 2 temperatures. One, 30 degrees, where we know H3K36 methylation is gone, but SPT6 is partly functional. Under those conditions, we see that there is a very good correlation between the level of antisense transcripts across the genome with CET2. So it seems reasonable to conclude that in an SPT6 mutant, this mutant at 30 degrees, the main reason we see elevated antisense transcription is because of lack of H3K36 methylation. However, when we compare them at 37 degrees, we see now that there is even more antisense transcription in an SPT6 mutant. And this suggests that now there's an additional defect that contributes to more extensive or higher levels of antisense transcription. We can also look at this in a different way, shown on the next slide. All right, here are gene body plots. All the genes in cerevisiae are listed from shortest to longest and from start to finish. So we're looking at antisense transcription going from right to left. The darker it is, the more transcription there is. And we're comparing wild-type strain to an SPT6 mutant. Here's the overlap. In future slides, I'm going to show you only the overlap. So the redder it is, the more transcription there is in the mutant. And you can see that there's extensive antisense transcription across the genome in this mutant SPT61004. We've also looked at this in a couple of other SPT6 mutants that seem to impair more specific aspects of SPT6 function. And I'm going to show you one of those on the next slide. This one's called SPT6YW. This is in the amino terminal, 5' part of the gene. And this affects specifically two interactions. In this mutant, SPT6 no longer interacts with histones, and it no longer interacts with that protein I mentioned, IWS1 or SPIN1. But the protein is still made. And you can see compared to what we view as complete loss of SPT6 function, or almost complete loss, there is a less severe effect on antisense, and it seems to be mainly around the 5' end. So something beginning in the open reading frames, going convergently to the normal direction of transcription. And you can see this on the metagene plots as well. SPT6 is in gold, wild type in green. SPT61004, we see an increased level pretty much across for antisense, whereas in the YW mutant, we see something that's more focused on the 5' end. So this makes us think that there are multiple classes of antisense transcription. There is a set2 dependent class that we see for this mutant at 30 degrees, and then perhaps at 37, in all of this, we're seeing this effect as well at the 5' end. So we think that there are multiple classes of transcription. Okay, finally, the other thing I wanted to mention, just in support of that, is that while SPT61004 pretty much abolishes H3K36 trimethylation, the SPT6YW mutant does not. It leaves it pretty much untouched. There are error bars here, just very small. Okay, now, I want to shift at the very end to another factor, SPT5. And just a word about SPT5. It's also conserved from bacteria to mammals, forms a complex with SPT4, otherwise known as DSIF. It directly interacts with RNA polymerase II. Here's a figure from a paper from Patrick Kramer's lab where they co-crystallized most of the 4-5 complex with Pol2, very specific interactions, and it co-localizes across transcribed regions. Even though it's just one number away from SPT6, it's viewed as playing very different roles in vivo. It's thought to affect, it's been shown to affect mRNA capping, transcription elongation in larger eukaryotes at the level of promoter proximal pausing in combination with the NELF factor. There's evidence that affects splicing and 3' processing as well. So we've begun to look at the role of SPT5 in transcription, sense and antisense, and we're doing these studies in POMBI. So we depleted the protein, as shown here. Here's before depletion, here's after, pretty much gone. This is using an auxin-inducible degron. And I'm just going to show you one type of analysis, and that's of antisense transcription, and that's shown here, the same kind of plot. And you can see in the SPT5 depleted case, there is, similar to the SPT6 allele, I'm going to put it here, there's an elevated level of antisense transcription across the 5' ends. So we're comparing RNA-seq to 5 depleted in POMBI to NET-seq to an SPT6 allele in cerevisiae. So hard to make a real comparison, but they both really suggest that there's this antisense transcription across 5' ends. So we conclude that both of these repress 5' antisense transcripts. So let me summarize here. SPT6 controls chromatin structure and histone modifications, controls many aspects of transcription, intragenic, antisense, and normal initiation. Many other factors play important roles in this regulation. We think that this analysis has revealed that the genome encodes more information at the level of both RNAs and proteins than we previously believed. And let me end with acknowledging all the people who did the work. This all started with Craig Kaplan and Lisa LaPrade many years ago. Vanessa Chung, Christine DiGennaro did all the work in POMBI. Magdalena Miroska did the NET-seq analysis. Steve Doris, TL-seq. These are people currently in the lab. Natalia Ream, Amit Shetty, Barack Alver is our bioinformaticist. He's a postdoc we share with Peter Park's lab. Scott Hallgren's another bioinformaticist who just joined the project. And Sterling Churchman, who we collaborated with for NET-seq analysis. And I will stop there. Thank you.
All right, questions for Fred?
Do you know whether the sites that you initiate transcription in SPT6 mutant, with the intragenic sites, do they correlate with hypersensitive sites?
They don't seem to correlate with anything that we know about chromatin structure, but we also have to remember that chromatin structure is greatly altered in SPT6 mutants. So we can try to compare wild-type chromatin structure to intragenic, but we don't really think that's a good comparison to make. We would really have to look at hypersensitive sites in the mutant.
Are there the same initiation sites in multiple experiments?
Yes. And I showed that when I showed the repeats for 2 different experiments for our TL-seq.
They start the same places in the mutant?
Yes.
Fred, beautiful talk.
Thank you.
So I'd like to turn the clock back a little bit and ask about SPT6 in the original TY delta screen. Because you described that what happens in those is that the delta transcript goes down and the HIS4 transcript goes up. So have you ever looked at that in detail in the SPT6? Perhaps in that one, they all go up, and therefore you can suppress the HIS4 phenotype, or is there truly in that one a reciprocal exchange of the use of the initiation sites? And if that does happen, how do you figure you actually are suppressing the delta while increasing the HIS4 in the SPT6, given the model for how it works?
So the short answer is that in that particular case, the longer transcript does not go down very much. We mainly increase the wild-type HIS4 mRNA levels. Consistent with that, when we look at intragenic transcription, at all the different ones we see, only in some cases does the longer one go down. In some cases, it still seems to remain at high levels. So we think that is particular to the different promoters that we're looking at.
So is there a connection between the elongation complex with SPT6 and some aspect of initiation as well? In other words,
Possibly. We need to look at that now, and that's especially highlighted by seeing effects on initiation at normal start sites. Rick.
Fred, it's interesting that SPT6 has that RNA-se domain in it, and I'm just wondering if you know anything about any possible functional role it's playing in detecting sense versus antisense transcription.
There's no evidence that it does anything. There was a careful analysis of that done by Chris Hill's group, and they went through motif by motif and actually showed that even though by primary amino acid sequence, many of them look like their motifs, when they look, there's often a residue or 2 that suggests that it probably doesn't have that function. And I'm pretty sure RNase-H was one of those. So there's no evidence that it does anything.
Hi, a question here. I was wondering about the correlation, if you would like to comment on the antisense transcription and the use of the internal transcription start site and the nucleosome structure change. The first 2 slides you showed the nucleosomes are more depleted for the transcription start site in your PT mutants than wild type, even more depleted over there. Is there some correlation over there?
We did an experiment a few years ago with Ali Rando's lab where we compared nucleosome levels across all the genes on chromosome 3 in wild-type and SPT6 mutants. And then we asked, is there a correlation between loss of nucleosomes and where there's intragenic transcription? And the answer was no. All right. However, that was before we had these data. We haven't gone back to look at it since. When we look at intragenic transcription by microarrays, we think we missed out on a number of them for various technical reasons. But we also think that we don't have to lose nucleosomes. It might be, there's some evidence that if you lose just K36 methylation, now there's going to be increased acetylation. That might also contribute to activation of those promoters.
Thank you.
So, Fred, following up that question, in your original flow 8 and sterile 11, you saw that there were Tata boxes that were regulating the start site. So, now that you see such widespread intragenic and 5', are they also regulated by Tata boxes? Are you
No.
getting normal?
No, if we take all the intragenic starts that we found and we ask, are there Tata boxes always there? No. They may be mildly enriched. If we just say in the neighborhood, 50 to 150 bases upstream, is there a consensus
It's
Tata?
just
Okay,
sloppy.
maybe a little bit above the 20% you might see, but not much more than that.
And is TBP at those? Are there?
We don't know yet. We're analyzing still some chip exodata with Frank Pugh's lab, and it looks like there are many more pre-initiation complexes in SPT6 mutants than in wild type. So there's some evidence that there are.
Hi, so how frequently does the antisense transcription actually affect the sense transcription mRNA in these SPT6 mutants?
We would really love to know the answer to that. We actually don't have strong data yet that the level of antisense affects the level of sense in a broad way. If we just do a correlation, when it's up, what happens to sense? We think that it's certainly true in specific cases, and many labs have studied a yeast gene, FO84, where they showed that the antisense controls sense, right? The Stutz lab and others. But there are some cases where it doesn't seem to happen. What we don't know here, of course, is if the antisense and sense are happening in the same cell or not, and what the relative levels are, things like that. So we really don't know the answer yet. There's no grand conclusion, though, that higher antisense means lower sense.
Okay.
So, Fred, your earlier POMBI work indicated that SPT6 might have a role in RNA processing, defects in small RNAs and other phenotypes you described. So how much of the increase in transcript that you described is actually transcriptional increase compared to defects in processing of those cryptic antisense RNAs?
In this work here in cerebricei, we think very little of it is processing, actually, because we see it by NET-seq, so we're looking at synthesis. We're not looking at processing or degradation. The antisense we see by NET-seq seems to correlate with what we see by northerns when we look. We haven't done RNA-seq, NET-seq comparison yet, but we really think most of it is at the level of synthesis, primarily based on NET-seq.
Fred?
Yes.
Nice talk.
Oh, hi, Brian.
Do you know if the antisense transcription is causing transcriptional interference with neighboring genes?
With neighboring genes, I'd say we don't know. We really don't know. There's no strong evidence of that. All we can say is that antisense transcription does not depend on gene configuration, but we don't know about with neighboring genes. The 5', that kind we've just begun to realize exists recently, so we haven't looked sufficiently. Yes.
Last question.
Yes. Hi.
Are you saying that SPT6 regulation about the antisense transcription is kind of set to dependent or independent? And for the scenario of the dependent pathway, I really compare, because SET2 just affect a limited number of the antisense transcription. I will compare the group of the genes.
I'm sorry, I didn't catch the question. If we compared what?
SET2 deletion just affects a limited number of the antisense transcription.
SET2, yes.
Yeah, have you really compared, the antisense transcription that caused by the SET2 and SPT6 were really correlated when the genes
well,
that affected?
we compared SET2 and SPT6 in terms of NET-seq results for antisense, and I showed that at one temperature where there's more SPT6 function that correlated quite well. When we deplete SPT6 more, then there's an increase in the SPT6 mutant. So we think it's partly due to loss of H3K36 methylation, but partly due to other defects as well.
Okay, thanks.
Okay.